Okay. कोई तो हाँ बोलो. ठीक है. Okay. Now, as you can see on the screen, right, the topic which I will be taking is enzymes. Now, this comes under, as you can see on the top side of the slide, right, it comes under paper 3 of botany for the semester 3, and my topic comes under unit 1. The name of the topic is enzymes, and in enzymes, you'll be studying 4 topics, as you can see on the screen. They are the purification and isolation of enzymes. Next one is biochemical regulation of enzymes. Next one is isoenzymes. And the last topic is vitamins or vitamins. You'll study their structure and their coenzyme activity. Hello. P. Okay. Now, as you can see, now when you talk about enzymes, enzyme study, you might have done a number of experiments with enzymes. Now, what are enzymes? As you are aware, they are also known as a biochemical catalyst, which take part, or which rather brings about an increase in the rate of the reaction of a number of biochemical processes. Now, when you consider the studies of enzyme or any experiments related to enzyme, you can either study in vivo or in vitro. Probably in your lower classes, you have studied some in vivo experiments with enzymes as well as with in vitro. Now, what is in vivo? In vivo experiments involve where you take the plant material or the animal tissue as it is and you try to study the enzyme activity without extracting it. That means within the tissue or within the cell itself. Now, as far as the plant, now when you consider the in vitro, probably you might have studied NR activity in your TY, wherein you have taken the either the what do you call as the radicals of the seeds, germinating seeds, or you can take leaf disc and you can study the what you can talk about the in vivo activity. Now, when you consider the in vivo activity, it has its own advantages. One of the advantages is that the activity of the enzyme is not lost as it is in its natural state, but it has its own limitations wherein you cannot find out where it is localized, and finding out the exact reaction may also be difficult. Nevertheless, in vivo studies are carried out for in case of plant, as I told you, root nodules, right? Sorry, we talk about the radicals of the root. You can take the entire radicals, right? And if you are talking about the enzymic activity, the source of the enzyme will be the radicals itself. And you put these radicals, or you can say leave this in a buffer and which contains the substrate. And after incubation, right, you can find out whether the activity, whether the product has been formed. Alternatively, you can also sometimes, if the enzymes are localized inside and they do not come out, then what happens is that you can take a section of the talk about the plant material, right, and you can study optically or under the microscope by carrying out various staining techniques, and you can localize where the enzymes are present. Now, in this particular chapter, as the name suggests, right, you will be talking about isolation and purification of enzymes. So we are naturally dealing with the in vitro, wherein the enzymes, we are not talking about the assay or the experiments, but rather we are talking about the isolation of the enzymes. Now, when you consider the isolation of enzymes or extraction, to a certain extent, even though, right, you have the topic isolation and purification of enzymes, isolation to a certain extent itself is a part of purification, because in isolation you try to separate the enzymes from a number of components. Nevertheless, we'll talk about the first topic, that is isolation and purification of enzymes, and this is what we'll try to complete today. Now, as you can see, it has been divided into two parts. Number one, isolation or extraction of enzymes, right? And the second one is purification of enzymes. Now, let us consider the first one, that is isolation or extraction of enzymes. Now, the very first most important thing, now as I told you in the beginning, right, you might have carried out certain enzymic experiments, right? Now, there are two choices for you. Either when you talk about a particular enzyme to be studied, either you know where the source is, or I say you have to extract and study the enzyme, particular enzyme from this particular source. So what does it mean? It means that you have a source known, and from that the source you have to isolate the particular enzyme and study and find out whether it is present or not, and find out how much is it present or what is its activity. Alternatively, you have a choice, right, where you say the source is not known. You have to just study the enzyme. Now, as you can see on the screen, isolation or extraction of enzyme, right? There are two components, right? One is the main important, as I told you, is a source. Now, source, it can be either specific, where you are told that this particular plant material is there. From this, you isolate the enzyme and study. Or alternatively, I just say that study the activity of a particular enzyme where the source is not known. Now, when the source is not known, you have a broad choice where it may be any plant material, it may be any plant part, whether it is root, stem, leaves, flowers, leaves, seeds, etc. Now, what are the case when you have an option? Now, here you try to take that source where the enzyme is found in plenty. Sorry, yeah, an enzyme is found in plenty, and where the isolation is possible. Now, the next part for isolation, or what you are supposed to know, is its localization. Where is it located? Whether is it located in the cytosol, or what you call it the cytoplasm, or whether is it located within the cell organ. Now, when you consider isolation, it will also depend upon the nature. When you talk about the nature, you talk about whether, right, the enzyme is free, that is soluble, or it is bound to membrane. Now, when you talk about cytosolic enzymes, right, usually they are free or soluble in nature. But when you consider those enzymes which are within the organelle, they may be either free or soluble within the matrix of the organelle, or they may be bound to the membrane. Similarly, when you talk about enzymes, they may be bound to the plasma membrane also. Now, let us move to the next part, that is extraction. Now, extraction depend upon the location, whether it is cytosolic or within the organelle. But then, as I told you, it can be either in soluble form or maybe bound to the membrane. So let us first consider, right, extraction of soluble enzymes. Now, when you consider the soluble enzymes, right, it is one of the cytosolic, as I told you, the cytoplasmic enzymes which are soluble. They are one of the easiest to extract. What did you do as far as plant material is concerned is that you take the plant material and just crush it, right, in a aqueous medium mostly, right, and the what you talk about the cell ruptures, and the contents, they are released out. You can carry out the filtration followed by maybe centrifugation if required, and you may get the source of enzyme. Next, as I told you, right, the soluble enzymes may be present within the organelles. Now, this is true for the eukaryotic cell. As such, they have the cell organelles within. Now, here, the separation of these cytosolic or cytoplasmic enzymes, the soluble enzymes present within the organelles, right? First of all, what happens is that it's not that easy, but at the same time, I won't say it is tougher, but then it requires or it involves a number of steps. Number one is you have to disrupt or break the intracellular membrane. How this is done? Most commonly, when you talk in the lab, you use what is called as a mortar and the pestle. Right? With the mortar and the pestle, right? You grind it, and due to this particular grinding and friction, the cell membrane and the tissue get ruptured, and the cell organelles are released. Right? Now, when you talk about the cell organelles are released, later on, as I told you, the required, now if you know where the enzymes are localized, right, by cell fractionation, right, or differential centrifugation, right, you can separate the cell organelles. And once you separate the required cell organelle, you can carry out the disruption or breaking of that organelle to release the enzyme. Now, what are the procedures? The general procedures involved in the extraction or isolation of the soluble enzymes. Now, as I told you, since we are dealing with plants, but then when you talk generally, right, the source may be an animal, maybe a microorganism, or maybe a plant. Now, in the case of plants, the organs are external, so it becomes you directly have the organs from which you can separate the enzymes. But as far as the animals are concerned, the organs are internal. So the animals, right, of interest has to be first sacrificed. And after sacrifice, right, the organ has to be removed, and from the organ, the tissue may be separated. In all these cases, whether it is plant, whether it is microorganisms or animals, right, care has to be taken. The isolated organ has to be kept cold. Since we are dealing with enzymes, and enzymes are affected by temperature, right, the organs or the tissue of interest has to be refrigerated. And also, the extraction procedure which will be carried out has to be carried under cold condition. One of the easiest, or what you do in the lab, is if you are talking about the mortar and pestle, it may be kept in the refrigerator, or you can have an ice bath surrounding the mortar, or if you talk about any other technique, right, of homogenization or grinding, or what are the case involved, right, you see that the surrounding temperature is maintained low, so that there is minimum loss of the enzyme activity. Now, as you can see here, as I told you, right, there are, you can isolate the enzymes from various sources, namely animals, plants, or microorganisms. Now, let us talk about animals. As I told you, on a sacrifice, the organs of the tissues are removed and they are kept cold, that is refrigerated to a temperature of about 4 degrees Celsius, right? And a number of techniques or procedures may be carried out for isolation of these soluble enzymes. Number one is the homogenization. Now, homogenization, as you may be aware, right, wherein it involves an instrument known as homogenizer. Now, in this homogenizer, what happens is the tissues, the soft tissues, right, they are crushed. And due to this crushing, right, the tissues get broken down, the contents are released, and after this, right, the what do you call as the matter can be, you can centrifuge to separate out the soluble components. Alternatively, technique is freezing or thawing. Now, what happens when you talk about freezing or thawing? Now, in this case, right, the tissue or the organ, right, is kept in a refrigerator, that too in a freezer, and because of this, right, the crystals are formed or becomes hard, and then later on, right, they may be crushed. Thawing involves, as I told you, freezing, and then later on allowing it to come to room temperature. And because of this also, right, the tissue, right, gets softer, much softer, and hence the homogenization or extraction of the enzymes becomes easier. Another technique for isolation of soluble enzymes is sonication. Now, sonication, as you may be aware, we have a sonicator in our lab. Now, what does it do? It produces waves. It produces